All right. I hope everybody had a good weekend and that you are all doing well. So I got a couple of announcements and then we will get started into thinking about T-cell effector function. First of all, if you were not here on Friday or if you didn't get one, I'm letting you guys bring an index card of this size, and I don't know what size this is, for the exam on Friday, and I have more of them up here if you need one. We'll have an exam on Friday. Hopefully that's not a surprise to anyone. We are discussing a paper on Wednesday, so note that that paper is posted, as are the questions for that paper. So be aware of that. I also sent you guys a doodle poll with times for a review session, and we still have a 3-way tie between 3 different times. For me, I feel like Wednesday works the best, but it's really kind of all over the place. My feeling is that way I can do it Wednesday and record it and you'll have it for Thursday, and that also means that I can just work on writing the exam on Thursday. We'll do Wednesday at 7 and I'll send you guys a Zoom link. It'll be on Zoom and I will record it. So that'll be the plan for that. So the plan for today is to talk about T-cell effector function. So basically what we are thinking about here is what happens after we've activated our T-cell. So once our T-cell has been activated with signal 1 and signal 2, and we'll see some more information about some cytokines today. Now that T-cell is going to be able to gain a function. And so at the beginning, it was a naive cell. It did come out of the thymus as either a naive CD4 or a naive CD8. It had made that co-receptor choice to determine if it bound better to class 1 in peptide or class 2 in peptide. But that cell did not have an effector function yet. It didn't have the ability to do anything about a microbe. Only after those signaling steps, some IL-2 and some expansion, will that cell actually gain its effector function and be able to do something in the periphery. And so today we're going to see those effector functions first of the CD8 cell and then of the CD4 cell. So when our naive CD8 T cell gets activated, it will become the activated form or the effector form of a CD8 T cell, which is a cytotoxic T lymphocyte or CTL. And so here we can see our CD8 T cell interacting with MHC class 1 for signal 1 and signal 2, and that CD8 T cell is going to become activated. It's going to proliferate, and so you can see that it's going to make lots and lots of copies of itself. And so now we have many little purple cells instead of just one, because we've told that cell, good job, we need you. You are responding to a microbe that is important. And that cell is now going to be able to induce effector function, specifically in the case of a CTL or cytotoxic T lymphocyte. It will be able to kill other cells that it finds that are presenting its peptide on MHC. And so you can see here that first activation of that naive T cell that's happening in a secondary lymphoid organ like the lymph node. And then later that cell is going to leave and go to the periphery and find other cells that are infected with that microbe and be able to kill them. And you can see that killing happening here. And I'll say some more about that on the next on the next slide. So one other thing, I feel like I didn't used to emphasize this as much as I do now, but it's something that came up in a lot of vaccine-related discussions. People were always very worried with their mRNA vaccine about getting the mRNA vaccine and having some of their cells potentially get killed that got the mRNA. The mRNA vaccine is allowing for activation of that first T cell to go from a naive T cell to an activated T cell. That T cell is only going to actually kill later after it's been activated and it finds that microbe. The first time that CD8 T cell sees antigen on, say, a dendritic cell, an APC, it's not actually killing that cell. It's becoming activated and gaining the function. It didn't have that function to begin with. And you can see, this figure as well. This dendritic cell is not getting killed. Instead, we're getting that activation part. And so only later, when the cell has gained its effector function, will we see that killing. But of course, it's all well and good for me to tell you that this killing happens, but it's more fun if I show you. So I'm going to show you a couple of example videos of this happening. And I've seen these videos a million times, and it took me forever to find links that went to the videos I wanted. So those are actually the videos that the links that I'm going to show you. So this is one version of it. The round cells are going to be our CTLs, or activated cytotoxic T lymphocytes, our activated CD8 T cells. And the long skinny cell is going to be our infected cell. It's actually technically a tumor in this case, but we're not dealing with tumors right now. We're just going to say it's an infected cell. And so you're going to be able to watch that interaction. There is a dye in the media of this experiment. And so if the membrane of the cell gets compromised in any way, so if the membrane is not sort of perfectly intact, the cell will take up the dye and will become fluorescent. And so what you can see happen, hopefully, maybe, besides loading, is that our cytotoxic T lymphocyte will come in contact with our infected cell and will, in fact, then induce apoptosis by giving a problem with membrane integrity. And you can see that those cells can actually detach and go attach to another infected cell to go kill that other infected cell. And that the CTL, the cytotoxic T lymphocyte, is not harmed by this process and can go and kill another cell. You can also see that, like, it's not as if like a bomb went off and every cell in the vicinity died. It was a specific killing of that specific cell. I'm going to show you one more video, Grace, and then I'll answer your question. So this is just one other version of this. The cytotoxic T cell is in purple. The infected cell is in blue here. And here we go. So you can see there's an interaction between the two. And then we go straight to the apoptosis of that infected cell. And there are a bunch of different kinds of dyes in this example, so I'm not going to go through all the things that they put dye in for this one, but you can see the general idea. So that's really what our cytotoxic T lymphocyte, or CTL, is doing with its effector function in tissues. Yeah, Grace.
How long does that take? Like, are those videos sped up?
They are sped up. Probably takes a couple hours. It's been a while since I actually have done experiments where, like, I timed it out, but that was a lot of years ago. So I feel like it was like a couple hours, but I'm not positive. One thing, again, you can notice here is that our cytotoxic T cell, our CTL, will interact specifically with one infected cell and lead to the death of that infected cell, but that other cells nearby are left alone. So it's not like we're going to destroy a whole bunch of tissue. It is a specific interaction and a specific killing of that one cell. You can also see that that CTL can, in fact, go and hit multiple cells. So if you have one activated CD8, that's a CTL, it can actually go from one cell to another and kill many different infected cells. Specifically, when the infected cell dies, when that infected cell is killed by a CTL, we are seeing a particular type of cell death, which is apoptosis. There are many types of cell death that one could talk about. Apoptosis is important for a couple of reasons. One thing to know about apoptosis, and it might be a little bit of a thing I get a little on a soapbox about, but I'll just tell you, apoptosis, sometimes you guys have learned it as programmed cell death. What that means, what programmed cell death means is that there's like signaling. It's not just like the cell just like explodes. Like there was a set of events and a set of specific steps involved in that death. And this figure is kind of trying to show you more of a less regulated cell death versus a regulated cell death by apoptosis. Just so you know, apoptosis is not the only kind of regulated cell death. There are other programmed cell death types. Apoptosis is just one of the programmed cell death types. It was the first one that was described. It's the one we know the most about. For a long time, people thought it was the only way of regulated cell death, but there's actually a lot of regulated cell death types. And when I think about apoptosis, one of the things that is really important, one of the ways that I think about apoptosis is I think about apoptosis as being very quiet cell death. Some of the other ones can cause a lot of collateral damage, a lot of inflammation. Apoptosis is just like the cell is kind of like, okay, I'm going to go away now and not disturb anyone else. That's apoptosis, unlike some of the other forms of cell death. That's important here. So as you can see it with this apoptotic cell, and you saw this in the videos as well, the apoptotic cell kind of shrinks in on itself and makes these little like packets, of stuff. It doesn't just shoot all its intracellular contents everywhere. It's important that our CTL is killing the infected cell by apoptosis, because the infected cell is, get this, infected. There's a microbe in that cell. We don't want to spray that microbe around everywhere. We kind of want to take all of the internal parts of that cell and close them off and not have them spray out any old place. And so that fragmentation, the making of these little bodies that can get cleaned up elsewhere, is a good way to make sure that you don't just spray virus all over the place like you might do with some other forms of cell death. So our CTLs are specifically inducing the target cell, the infected cell, to undergo apoptosis. There are two ways that a CTL can make a target cell undergo apoptosis. One of them is using a protein called Fas. So you can see that the CTL has this protein called Fas ligand on its surface, and Fas ligand can bind with a protein called Fas on the surface of our target cell, our infected cell that needs to be killed. And if we have this Fas ligand Fas interaction, that can induce apoptosis of our target cell. And so we can see this kind of protein-protein interaction between a surface protein on the CTL and a target cell leading to apoptosis. You can also see how this would be pretty specific. The CTL isn't just killing everything around it, it's killing the specific cell that it touches with its Fas ligand. When Fas in the infected cell is ligated by Fas ligand, we get this type of apoptosis pathway happening. I'm going to hit a couple of highlights here, and a lot of this is for compare and contrast later. Usually, when apoptosis happens because we've turned on a protease called caspase 3. And caspase 3 cuts off stuff. And that induces the cell to die. So usually caspase 3 is pretty key in making apoptosis happen. If our cell has Fas on the surface, and that Fas gets triggered, we can trigger some other molecules above caspase 3 to become activated. One of them is called caspase 8, so we can turn on caspase 8, and you can see caspase 8 can turn on caspase 3 and give us apoptosis. Some other parts of apoptosis include some proteins that happen or that are involved in the mitochondria. Caspase 8 can also activate some of that mitochondrial pathway, and eventually that gets us to caspase 3 as well. So this is how this can look with Fas on our infected cell if the Fas ligand from the CTL tries to interact with and kill that cell. Like I said, Fas ligand is one way that cytotoxic T lymphocytes can kill infected cells. The other way that this can happen is due to something called cytotoxic granule release. And so the cytotoxic T cell, the CTL, has some special granules inside it. There are these little red packages that are shown here from your textbook, and you can even see them as these little red dots in this green cell microscopically. They're listed here, that they're labeled there as LG, or lytic granules. Lytic granules, cytotoxic granules, it's the same thing. But these granules contain products that can kill another cell. So these are sort of toxic products that are going to kill other cells. And so when we have our CTL interacting with the target cell, you can see we've got T cell receptor interacting with MHC. Remember I told you about the integrins that are kind of holding this whole thing together and also making a little wall. The T cell can actually secrete those cytotoxic granules right here at that site where the T cell receptor is, because it knows that's where the bad guy is right there. And those cytotoxic granules will sort of go across the cleft between the two cells and will induce apoptotic cell death of this target cell. And they won't go just all over everywhere because the integrins are kind of making a wall. And the cell isn't secreting in every direction possible around itself. It's only secreting in this directional fashion right at that other cell. And so here in this microscopy image, you can see now our CTL. It used to have cytotoxic granules sort of everywhere. Now it's rearranged things so the cytotoxic granules are right at the contact site with that infected cell. And eventually this T cell is going to secrete stuff to the target cell to induce apoptosis. And so you can kind of see this as being like the toxic bomb release. And you can also see this process here. Oh, look, Grace, it even has times on it. So you can see our CTL coming into contact with the target cell. You can see it kind of rearranging to put all those granules by the target cell. Then it will release those granules just at the target cell, not kind of every old place, because you don't want to destroy all the tissues. And then it's going to leave, and this target cell is going to undergo apoptosis. The TTL might be able to go find another target cell. And so so we might wonder at this point, what was in those lytic granules or those cytotoxic granules that allowed the CTL to kill the target cell? In reality, the answer is a bunch of stuff. But I'm going to tell you about two things that are in there that are really important things. One of them is a protein called perforin. If you hear the word perforin, what does the word perforin remind you of? Yeah, Vicky. Perforate. What's perforate mean? Make holes. Guess what perforin does? Perforin makes holes in the membrane of that target cell. And so here you can see a granule full of little perforins, and you can see that the cell might release that granule, and these little perforins will interact with the membrane of the target cell and make a hole in the target cell. And so now, stuff that's supposed to stay inside the cell leaks out, and stuff that's supposed to go out stays in. You can imagine how this is pretty easily going to induce cell death here. And you can see some pictures of perforin results on a cell membrane on the right. If you look at that image, does that image remind you of anything? Yeah. The MAC, remember the MAC from complement? Remember the membrane attack complex in complement? Looked pretty similar to that. They look so similar that you can't actually distinguish photographs of them. And honestly, I swear in every textbook, they just pick a picture and tell you it's MAC or tell you it's perforin, and they look so identical. So it's the same idea of what we're doing with the innate immune response of complement, of breaking holes in the membrane. Here we're breaking holes in the membrane again. So very similar process. The other thing about perforin, however, is that perforin can also make these holes that let some of the other things that were in the granule into the cell. So not only is perforin just perforating the membrane, and that's bad in and of itself, it's also letting other stuff that was in this granule go into the cell. And so you can see that here. Here is the granule. It might have a bunch of different things in it. Perforin, the green cylinder, as well as other stuff. Perforin can make a hole. And that also can let the other stuff into the cytoplasm. And again, there's a lot of other stuff. But the most famous of that other stuff is called granzyme B. The end of granzyme sounds like what? What's it? Enzyme. So in fact, the word granzyme is granule enzyme smushed together, because it was an enzyme that's in the granules. So what you can know about granzyme B is it has some enzymatic activity, and it was in the granule. And specifically, the enzymatic activity of granzyme B is that it cuts up stuff. Specifically, it cuts caspase 3. So it just gets rid of the middleman, goes straight to the bottom of that apoptosis pathway I was telling you about, and cuts caspase 3. It can also cut stuff in the mitochondrial part of the pathway to amplify, but really the idea here is that we just go straight to the bottom, cut caspase 3, get the cell into apoptosis. So perforin has both this sort of membrane permeability and this let-in granzyme function. There are other things in the granule. I'm not going to get super worried about them right now. I will just also tell you that cytotoxic T lymphocytes are also pretty famous for their production of interferon gamma in order to induce some additional immune responses. And we'll see more interferon gamma today, but interferon gamma is a pretty famous cytokine that CTLs make as well. And so besides making all of these cytotoxic molecules, interferon gamma is another big thing that cytotoxic T lymphocytes make. Interferon gamma is not one of the interferons we saw earlier in the semester. Earlier in the semester, we saw the type 1 interferons, alpha and beta. Interferon gamma is a type 2 interferon, which is involved in adaptive immune responses, like CD8 T cells, instead of the innate responses like we saw earlier. So those are the details of our CD8 T cells. So once our cell starts out as a naive CD8 T cell, once it's been activated, it will differentiate into a CTL, and it will be able to kill target cells. We can also talk about the effector function of CD4 cells. And in some ways, effector function of CD4 cells is like super simple to explain, and in some ways it's less simple to explain. You can see it's happening at the top of this slide along a very kind of similar type of process that we saw with the CD8s. And we typically will call this CD4 T cell that has gained effector function a helper T cell. So it starts out as a naive CD4 cell, gets activated, gains effector function, and becomes a helper T cell instead of a cytotoxic T cell. And so helper doesn't seem to be as useful of a term as cytotoxic, because you're like, what does helper mean? What's helping? And the answer really for a helper T cell, you can see I've got T with a sub H at the top here, which is a way that people often write helper T cell. The point for helper T cells is that they make cytokines. And so really what a helper T cell means is a T cell, an activated CD4 T cell that's going to make a bunch of cytokines. Those cytokines are stored often in little granules, and those granules get concentrated at the site of contact with the antigen presenting cell and get secreted across this little juncture, right where those same integrins are, with the same wall, and you get the same kind of directional secretion of that cytokine. So in that way, it all looks really similar. But what you will notice here is I have not named the cytokine. I just said the helper T cell makes the cytokine. And the reason for this is that there are different kinds of helper T cells. So there are different kinds of CD4 cells or activated CD4 cells, and each one makes a different kind of cytokine. And so as we're going to talk about these different types of helper T cells and their different cytokines. In thinking about this, you might think about how the cell decides to be one of those different types of CD4 T cells. So remember, we saw a little bit about how the cell decides to be a CD4 versus CD8 in the thymus. Now, if it's a CD4, it has more choices to make. It has to choose which kind of CD4 cell it's going to become. And most of the time, yes, we'll get to the exception to that later. Most of the time, that decision is made in the periphery. And that decision is made based on how that cell got activated as a naive cell. So basically, the idea is that the events that happen right here in activating this naive CD4 cell also push that naive CD4 cell to become a specific, which is not the same word as particular, and if you try to say those two words together, it's really weird, a specific type of CD4 cell. All of this is based on what kind of cytokines that CD4 cell sees when it first gets activated. So with CD4 cells, the cytokine, that signal 3 cytokine, is super important and can push that CD4 cell down one of a bunch of different paths. So exactly what cytokines that original antigen presenting cell is making and providing for signal 3 as we activate our naive T cell will turn that naive T cell into an effector of one of many different kinds. As I'll say a little bit more about, and this might also get a little bit to me age-wise. When I sort of started in immunology, there were 2 kinds of CD4 cells. And now there's a lot more. And when I was in graduate school, it was kind of at the time where we were discovering a whole bunch of them. And sometimes it felt a little ridiculous. Sometimes it was like, oh, these are the CD4 cells that respond to fungal infection in your left pinky on Tuesdays. And it was kind of like, okay, guys, chill. Like, we don't need this many subtypes. And so there became sort of this set of rules of how, like, a subtype was, like, a real thing. And those rules are kind of going to be important for us to be thinking about. And so what we have realized is that for any particular type of T cell to get pushed to into a specific kind of subtype, it quote unquote counts as a subtype if we can name 3 specific things. And so I tell you this because for all the subtypes I'm going to be teaching you about, the key thing is you're going to need to be able to name those 3 pieces about each subtype. So one of the things is, what cytokine does the cell need to receive during its activation? Or, we often refer to that as the polarizing cytokine. Which cytokine tells the T cell, go down this path versus go down the other path? And so we need to be able to identify what is that cytokine. What we have learned is that when one of those naive T cells gets a particular cytokine, when that T cell receives its polarizing cytokine, that turns on a unique transcription factor. And so there's some kind of transcription factor that gets turned on. Here it's called a master gene regulator. I would also be fine calling it just a transcription factor. And so one of the cell types we're going to hear about is one called Th1. And so basically there are certain things that a cell does when a CD4 T cell does when it's a Th1 cell. And the thing that controls all of the Th1 functions is this transcription factor. When you start making this transcription factor, you start living the Th1 life. And the reason why you know, oh, it's time to make that transcription factor is because you got this original polarizing cytokine. So this cytokine leads to that transcription factor. Then when you make that transcription factor, that induces you to do certain functions. In particular, it tells you this is what cytokine you make. Because you chose the Th1 life, this is the cytokine you're going to make. And so we also have to know what cytokine does that subset make. What's its sort of defining subset. What I hope that you can notice here is that this is another place where we see adaptive immune responses and innate immune responses tightly interlinked. Because our initial antigen presenting cell, our initial cell that's turning on the T cell, could make different kinds of cytokines, and that can push the T cell down different paths. And if we can think about, well, why would the dendritic cell, say, want to push a T cell down one path or another? Because that's kind of what it's doing, right? And the answer is, well, it has to do with what PRR got stimulated at the beginning. So, for example, if there was a PRR that is from a fungus, that's going to make the dendritic cell make one kind of cytokine, and that's going to push the T cells down one path. And oh my gosh, the path that the T cells get pushed down is the best possible path for dealing with fungi. So it's like linked. If we see certain types of parasitic worms, we might push the T cell down another path. And it just so happens that path is a really good path for dealing with parasitic worms. And so on. And so all of this is kind of related to the innate immune system controlling, I saw this kind of pathogen, I'm going to push T cells towards the correct type of response for that pathogen. Sometimes people talk about the helper T cell or the helper T cell, let's go with that. Sometimes people like to talk about it as the quarterback of the immune system because it sort of pushes the immune system into different directions based on which cytokine that's making. And if that's the case, then like the dendritic cell is the coach that tells the quarterback which direction it should go based on those PRRs. So here are some of our helper T cell subsets. And so you can see that our naive T cell can become any one of these different helper T cell subsets based on which cytokine that naive T cell receives at its first activation. So all the CD4 cells start out as naive, but they get pushed one direction or another with cytokines. That's going to change which transcription factor is made, and that's going to change the function of the cell and the cytokines that it produces. I'm not going to talk about all of the subtypes that are shown on this slide. But what I like about this slide is it does do a really good job of putting polarizing cytokines, transcription factors, effector cytokines, and why you care all in one place, even though it does show more subsets than I'm going to be telling you about. So don't freak out by all the kinds of cells here. We're not going to get quite this many. But I like that we've got kind of everything laid out nicely on this slide. And this is the similar version from your textbook. And in fact, really, this is sort of these are the subsets that I'm going to tell you about. One thing I also want to point out to you is that in some cases, the cytokines that I'm going to tell you to induce differentiation and the characteristic cytokines are going to be like really clear. There are a couple of other ones where there have been some arguments over the years, and I think different slides say slightly different things. When you are answering questions about this on the exam, if it's on the slides, it counts as correct, even if it's not exactly the ones that I told you. Though I will point out there are going to be some details about TGF-beta that are going to be really important, and you can't mess that one up. Okay? So sort of what's going on with these ones that include TGF-beta, we're going to need to care about. But in particular, there are a couple places where I know the two textbooks I use figures from don't agree. And I'm flexible on what you put on for answers for those. So at the beginning, there were just two. We first thought there were just two. And we called them Th1 and Th2. Helper T cell number 1 and helper T cell number 2 types. And so you can see kind of this general idea of Th1 versus Th2 activation here. And so what we know about Th1 and Th2 cells is that for Th1 cells, I think I like it this way better. Th1, the polarizing cytokine is IL-12, and it's always IL-12. When a naive T cell gets IL-12 during its activation, it turns on a new transcription factor. And that transcription factor is called TBET. Once the cell starts making TBET, that tells the cell how to do Th1 things. And the most important Th1 thing is that now Th1 cells make a particular cytokine. And that cytokine is interferon gamma, which you saw a little bit earlier. And so interferon gamma is going to help the rest of the immune response in a particular way. Because remember, the whole point here is a helper T cell. It's going to make some kind of cytokine to help the rest of the response. And alternatively, we have Th2 cells. Th2 cells are polarized. Originally, it was described as IL-4. And IL-4, still, if you write IL-4 here, you're good. Some people might get a little confused here, and so I will say that we now think that another cytokine called IL-33 might actually be the answer. But if you say IL-4, it's okay. If you're confused and don't like IL-4, go for IL-33. When the cell receives these cytokines, that makes the cell start to make a new transcription factor, and that transcription factor is called GATA3. And when the cell starts making GATA3, now it starts transcribing Th2 things. And Th2 things, most famous is IL-4 again, but really IL-4, IL-5, and IL-13 all kind of come together as a package deal. The reason why some students will get confused is they're like, wait, IL-4 makes it make IL-4, and it feels circular, and they're like, but for the IL-4 in the first place to make the other IL-4. So that's why we're sort of, we kind of think IL-33 is related to this now. And so you can see this same process here with your Th1 cell. So here you can see your Th1, your naive T cell can get activated in the presence of IL-12. That will induce that cell to start making TBET. And now that cell will start making interferon gamma. The thing that's really important here, and so you can see that in various ways, is why the heck do we care about interferon gamma? What is interferon gamma good for? What's it going to help your body do? And interferon gamma can do two really important things for us. One thing that interferon gamma can do so I guess results, is that interferon gamma can enhance the ability of CD8 T cells to do their job. It's also going to enhance a cell type called an NK cell that we'll talk about later on. So basically, we told CD8 T cells, like, get to work and get good at working. We also will activate macrophages here. And the idea with activating macrophages is that sometimes macrophages may have some things inside of their granules. And if the macrophage gets activated, that helps the macrophage better kill stuff in its granules. So that macrophage does more respiratory burst, kills, like, makes more enzymes to kill things in the granules, all that kind of stuff. So we now start to activate that macrophage, kill more things. And so what you might notice with both our activation of macrophages and our activation of CD8 T cells is that we are doing a really good job here of getting rid of intracellular pathogens. So like pathogens that live inside of cells. Because the CD8 T cell is going to kill pathogens inside of cells. And an activated macrophage is going to kill things inside of cells. So Th1 is really good for intracellular pathogens. Particularly intracellular. Th1 responses, when they go wrong, can also be related to a number of different kind of autoimmune diseases. So we can also see autoimmunity if this goes incorrectly. Alternatively, we see Th2 cells. And as you can see, Th2 cells are polarized by receiving IL-4. They start making GATA3, and then they start making IL-4 and some other cytokines like IL-15 and IL-13. This set of cytokines will help other parts of the immune response. So now we're not going to turn on CD8 T cells. We're not going to turn on macrophages. Instead, we're going to turn on a different sort of lane of the immune system that might be ideal for a pathogen that's going on here. And so some of the functions of our Th2 cytokines are shown here. One of them is that we really turn on eosinophils and mast cells. So we're going to instead of turning on CD8 cells and K cells, we're going to really turn on eosinophils and mast cells. We are also going to induce some antibody production, especially IgE. And we may also kind of increase mucus secretion and peristalsis and basically body functions that expel things. You can see some of the details of mast cell activation happening here. Again, we've got a bunch of granules that can fuse with the cell membrane and secrete toxic products. But now they're specifically secreting toxic products sort of at a microbe. And Th2 responses are particularly good at dealing with things like parasitic worms. And if you think about these functions like eosinophils degranulating, mast cells degranulating, expelling stuff, all of those things are going to be really, and even antibody production, all of those things are going to be really useful for a pathogen that's outside of a cell, not inside of a cell. And in particular, it's really useful for extracellular parasites, like parasitic worms, so like a eukaryotic organism. You just want to throw everything out. And you want to throw histamine at that worm to try to kill it. And so here we're going to see a really nice response to extracellular parasites. I ran out of room. And so you can see that these two responses are kind of, acting against slightly different sets of pathogens, are useful in different situations. There's one other really important, a couple other really important pieces to understand about the control of Th1 and Th2 cells. So this is sort of a general overview. You can see an intracellular pathogen induces our antigen-presenting cell to make IL-12. Now we're going to induce our naive T cell to become a Th1. It's going to make Tbat. See, now it became a Th1. I don't know why it got smaller. And it's going to make interferon gamma. And interferon gamma is really good for our intracellular pathogen. While if we have some kind of extracellular parasite, that's going to induce our dendritic cell to make IL-4. It's going to polarize our naive T cell to make GATA3 and then make more IL-4 as a Th2 cell. And that's going to be really useful in getting rid of extracellular parasites. Hooray! Sometimes different versions of these books will talk more or less about Th2 and antibody production. You can see I kind of said some IgE and then I just kind of floated over antibody production. One reason for that is that there can be antibodies in both of these situations, but the specific subtypes of antibodies will be different. And so if we look at, say, interferon gamma, we can see that if there's interferon gamma around, we're going to see some types of antibodies like IgG3 and IgG2A and less of others. While if we see IL-4, we're going to see a lot of IgE and IgG1. So I don't love the idea of calling one of them the antibody one, because really they're all pushing antibodies in slightly different directions. The other thing that we have to know about Th1, Th2 cells leads to, so many places. I'm going to say it now. I used to, in past years, go down one of the rabbit holes here. I'm saving the rabbit hole for later. But you guys might have heard things about if you aren't exposed to enough microbes, if your life is too clean, you'll have allergies and things like that. If you heard those kind of ideas. That is actually based on a piece of Th1, Th2 biology. It's related to something called the hygiene hypothesis. We're going to talk about it more later. I'm not going to get way into it right now. But one piece of it that is related to is the fact that there is feedback between these two types of cells. And so I told you that IL-12 turns on Tbat and that Tbat turns on interferon gamma and gives us a Th2 response. However, IL-12 also inhibits GATA3. It turns off GATA3. And IL-12 and Tbat turns off GATA3. Not only does Tbat turn on interferon gamma, Tbat also turns off IL-4 and IL-5. So once a cell starts getting pushed in the Th1 direction, there's sort of positive feedback for more Th1 and negative feedback to say no Th2. Similarly, IL-4 turns on GATA3, but also inhibits Tbat. And GATA3 is inhibiting Tbat, GATA3 is inhibiting interferon gamma. So again, not only are these cytokines promoting their own kind of helper T cell subset, they also are inhibiting the other kind. So once you kind of start making one response in one direction, it just kind of builds and builds and builds more and more in that direction. So you tend to, if you're responding to a particular pathogen, you tend to make a Th1 or a Th2. And for genetic reasons, some people are predisposed to do one or the other. Like I personally am predisposed to a Th2 response. So I am more likely to make a Th2 response than a Th1. And once I start making a Th2, it's going to keep going and going and going and going down the Th2 path. And then I'm going to have allergies and I'm going to be angry. But, so know that there's a lot of feedback between these. So this was where kind of helper T cell biology was when I was getting close to finishing graduate school. But as I said, we have since realized that there are more than just Th1 and Th2. And I'm going to tell you about kind of the next big one. And in some ways you might look at this and be like, wow, I learned with complement that immunologists can't count, but now they really can't count. Cause you kind of want the next one to be called Th3, and it's not. But it's okay, because you're going to end up liking what it's called by the end. And so the next one that I'm going to tell you about, there was Th3 for a while, but we don't usually call it Th3 anymore. And it's another name, and it'll be happening later. The one I'm going to tell you about next is called Th17. And the reason why Th17 cells were named Th17 cells is because they make IL-17. So the idea was we're just going to name them Th17 cells because their job is to make IL-17. These cells make IL-17 because of the transcription factor that they've turned on. And that transcription factor is called ROR gamma T. And so we can have ROR gamma T be made that induces the cell to make IL-17. And in order to push a cell to become a Th17 cell, we need a combination of cytokines. And so note that I'm pretty chill about what you write here, but in fact it has to be a combination. It needs to be a combination. Usually I think about it as a combination of TGF-beta and IL-6. And that IL-6 is going to be particularly important here. You've heard of IL-6 briefly earlier this semester. So where have you heard of IL-6 briefly earlier this semester? Yeah, Josh?
Was it one of the cytokines that was related to inflammation?
It, in fact, is an inflammatory cytokine. It's made by the innate immune system in situations of inflammation. It's involved in like fever and stuff like that. So the key thing here with Th17 is you need inflammation plus TGF-beta. And so yeah, it varies in a couple places of exactly what's going on, but it's inflammation, like IL-6, plus TGF-beta in combination with each other. IL-17 is particularly good at turning on neutrophils. And what we have learned about IL-17 is it's really good at dealing with extracellular bacteria and fungi. So things that, Pac-Man can just come along and eat up. So it's extracellular bacteria and fungi. It's also here. Extracellular bacteria and fungi. Or even here. Extracellular bacteria and fungi. And so that's really the sort of the key piece of Th17. Th2 responses are really important in allergy and asthma. Th1 and Th17s tend to be particularly important in some kind of autoimmune diseases. So that's my simple Th17. One of the more recently described types of T cell subsets are known as the TFH cells. So here, we just gave up numbers altogether. And instead of telling you about cytokines or numbers, the abbreviation tells you something about what the cell does. So TFH cells help B cells in the lymph node or in the secondary lymphoid organ follicle. So they help B cells specifically in a place called the follicle. And so the TFH are the T follicular helpers because they're doing helping in the follicle. TFH cells are really critical for making a good B cell and antibody response. If you remember when we were kind of following the life of a B cell, we said, and then it goes to the periphery. And then I can't tell you anymore. And the reason is because all the rest of the events in the B cell's life include a TFH, a T follicular helper cell. So that's why actually we come back and sort of our next thing we talk about is what happened to that B cell. Because now we've got a T follicular helper cell to do stuff with it. Specifically, that T follicular helper cell is going to make a cytokine called IL-21 in order to help those B cells. It is going to produce a transcription factor called BCL6. And you can see that IL-21 or IL-6 can polarize a T follicular helper. But notice this IL-6 is different. This IL-6 is not in combination with TGF-beta. So the IL-6 by itself might push T follicular helper, but the IL-6 in combination with TGF-beta is going to push Th17. And again, the idea here is that this is really going to help B cells and help induce a good antibody response. And so you can see a T follicular helper cell's main job here, which is that a T follicular helper cell is going to be providing help to a B cell so that that B cell can become fully activated. So these are basically specifically helping B cells. This is the other reason why I don't like calling any of these the antibody one. Because in some ways you're like, well, shouldn't this be the antibody one? It's helping the B cell, and in some ways it kind of is. So you can see that. So those are 4 of our types of cells. And I was going to want to tell you about 5. I'm going to start telling you a little bit about the 5th one. And we'll finish talking about that 5th one a little bit on Wednesday, because I'm not going to have enough time to talk about it. But we're going to see it more on Wednesday after we talk about the paper. The 5th one was actually the 3rd one described. It was Th3 for a little while. And it is unique. Well, it's unique for a bunch of ways. Its function is super, super, super important. If you don't have these cells, real bad things happen. But this is also unique because unlike the others, there are 2 ways you can make this cell. So all of these other ones, it's sort of a very straightforward process. You have a naive cell in the periphery. The naive cell receives a polarizing cytokine. That cell turns on a transcription factor. Now we make the effector cytokine. There's still going to be a transcription factor. There's still going to be all this stuff. But there's going to be 2 totally different pathways to turn on this final set of T cells. This final set of T cell is called the Treg, or regulatory T cell. And the job of a regulatory T cell is to turn off other types of immune cells. So it basically tells everybody else to get turned off. If you don't have Tregs, you have massive autoimmunity. In fact, Tregs are another big part of peripheral tolerance. And so you can see Tregs are specifically acting to turn off other cells. For a long time, or not for a long time, there was controversy. Remember how I told you last time there are two words that when I say them, I think I'm going to get struck by lightning? When people talked about Tregs, there used to be this kind of cell called a suppressor T cell. And then suppressor T cells became very out, and nobody likes the word suppressor T cells. And so suppressor or suppress is another word I can't say without getting struck by lightning. So you can imagine a Treg basically suppressing other immune cells. Immunologists just really don't like the word suppress. If you ever hear regulate, immunologists say this regulates blah, blah, blah. They usually mean suppress. They just didn't feel like saying the word suppress because they were scared of it. And so regulatory T cells are kind of what suppressor T cells turned into. And they act by turning off other cell types. And we'll talk about ways that we make regulatory T cells and exactly how they suppress other cell types on Wednesday, along with talking about our paper. So I will see you guys on Wednesday. We will have our review session on Wednesday, and I will email you about that with a Zoom link. Please get an index card if you don't already have one.